day everyone, I'm Hailey Casini Navarro, and for today, I'm going to explain to you what is mitochondria. These are the small parts found inside almost every cell in our body. They are called the powerhouses because they're the ones that gives energy to the cell. Pag waray mitochondria, cells would not have enough energy to stay alive. I'm going to show you a video clip on how mitochondria produce energy. Our body is made up of trillions of cells. They all require energy to function. This energy is created within ourselves in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane separating it from the cytosol and an inner membrane surrounding the so-called matrix. The area between this membrane is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, or the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP. And that is how mitochondria produce energy. And now, let's discover the important parts of mitochondria.
What is the outer membrane? The outer membrane usually refers to protective lipids bilayer found in certain cells and organelles. In gram-negative bacteria, it's the outermost layer of the cell enveloped outside the peptidoglycan cell wall. Let's have the composition of in gram-negative bacteria. First, phospholipids, the inner leaflet. Second, the lipopolysaccharides LPS, the outer leaflet, important for structural integrity and pathogenicity. Third, purines, protein channels that allow passage of small molecules. Let's have the functions of in gram-negative bacteria. First, acts as a barrier against antibiotics, detergents, and toxins. Second, provides structural strength. Third, plays a role in immune system recognition and toxin activity of LPS. In mitochondria, the outer mitochondrial membrane encloses the organelle. It contains porous proteins, BDAC, that make it permeable to small molecules and ions. It regulates transport of proteins into mitochondria through the TOM complex, transclocase of the outer membrane. In chloroplast, similar to mitochondria, chloroplasts have an outer membrane. It is relatively permeable due to purines, allowing small molecules to diffuse in. Works with the inner membrane to regulate transport. Let's have the explanation of outer membrane. The outer membrane is a lipid bilayer that surrounds some cells and organelles, acting as protective barrier and controlling transport. In gram-negative bacteria, the outer membrane lies outside the cell wall. It contains lipopolysaccharides, the LPS, which protect the cell can trigger strong immune responses. It has also purine proteins that also channels for small molecules. It means it protecting bacteria from antibiotic, toxins, and harsh environment. In mitochondria, the outer mitochondrial membrane covers the organelle. It has pores that make it permeable to ions and small molecules. And it contains special protein complex like TOM that help important large proteins. In chloroplast, the outer membrane surrounds the organelle and fairly permeable, allowing small molecules to pass while working together with the inner membrane to regulate entry of large molecules. In short, the outer membrane is a selective protective barrier found in gram-negative bacteria, mitochondria, and chloroplasts that helps control what enters and exits while providing structural defense. That's all. Thank you.
Moving on to the inner membrane. The inner membrane of the mitochondrion is one of the most important structures in the cell. Unlike the smooth outer membrane, the inner membrane is highly folded into shapes called cristae. These folds are like wrinkles that increase surface area, just like how folding a towel gives more layers in the same space. The cristae give the mitochondrion more room to perform chemical reactions. Inside this inner membrane, there are special proteins and enzymes that carry out the electron transport chain and use ATP synthesis to produce ATP. So ATP is often called the energy currency of the cell because it powers almost everything our body does, from contracting our muscles when we walk to sending signals in our nerves and helping our organs function properly. The inner membrane also creates two compartments, the intermembrane space and the matrix. By separating the spaces, it can control the movement of protons, creating what we call a proton gradient. This gradient is like building up pressure behind a dam. Once released, it generates energy that the cell captures as ATP. So for example, every time we eat food like rice or bread, the sugars are broken down, and the mitochondria use the inner membrane to convert those nutrients into ATP. So without this process, our cells wouldn't have the energy to keep us alive. It is also very selective. It only allows certain molecules to pass through, making sure the right materials enter the matrix. This control is like a security guard at a concert gate, checking that only people with tickets can go inside. So overall, the inner membrane is not just a wall. It is the real power site of the mitochondria, where the most important steps in energy production happen. Without it, our cells and even our entire body would run out of power, just like a phone with no battery. And here's the sketch of the inner membrane. First is the outer membrane, is here. The outer membrane acts like a protective cover that allows small molecules to pass in and out of the mitochondria. Next is the intermembrane, the small space between the outer and inner membranes where protons build up to help make ATP. Then the inner membrane, folded membrane that increases surface area for energy production. This is where the electron transport chain happens. These little dots here are the ATP synthesis, an enzyme located on the inner membrane that uses the proton gradient to produce ATP. And lastly is the matrix, the innermost space of the mitochondrion where enzymes, mitochondrial DNA, and the Krebs cycles are found. That's all.
The main parts of the mitochondria. The fold inside that increase the working area so the mitochondria can produce more energy. In mitochondria, cristae junction or cristae function are tubular or a narrow connection between the inner membrane and the cristae. So why mitochondria are important? Because it can increase the surface area of the inner membrane, allowing for more efficiency through the cellular respiration. Cristae are vital for maintaining cellular energy homeostasis and overall cellular function. Cristae junctions help to maintain unique structure or function of mitochondria, which are essential for energy production in cells.
After that, let's proceed to the matrix. Kung mani-explain na ako kung ano ang matrix. So, the mitochondria is often called the powerhouse of the cell because it produces energy. But inside it, the matrix plays a very big role. So, ano nga ba ang matrix? The matrix matrix is the jelly-like fluid found in the innermost part of the mitochondria. It may look simple, but it's filled with important materials such as enzymes, DNA, and ribosomes. Now, why is the matrix so important? Because this is where food is broken down to release energy. Inside the matrix, a special process called Krebs cycle or citric acid cycle happens. This is like a step-by-step system that takes the nutrients from the food we eat, like sugars and fats, and turns them into energy. The energy produced is in the form of ATP, which stands for adenosine triphosphate. ATP is what we call the energy currency of the cell, meaning it is the main source of power that the body uses for everything. Kung wala ang ATP, our muscles cannot move, our brain cannot send signals, and even our heart would not be able to beat. To make it simple, you can imagine the matrix as the factory or workshop inside the mitochondria. The food goes in, it gets processed, and the finished product is ATP, the energy that keeps us alive and active. So, to conclude, the matrix is not just a pillar inside the mitochondria. It is the place where the most important job happens, turning food into usable energy. Without the matrix, our cells and even our whole body would not have the energy needed to survive.
Hello everyone, I am Rian Trisha and I'm here to discuss about mitochondrial DNA. So, what is mitochondrial DNA?
Mitochondrial DNA is a small circular genetic material found in the mitochondria that is inherited from the mother and is responsible for helping produce energy in the cell.
Let's deepen our understanding with this video.
Mitochondrial DNA. Mitochondria are structures inside the body that are responsible for energy production. They contain their own DNA and replicate independently from the cell. Mitochondria are passed through a mother's egg directly to both her sons and daughters. In turn, her daughter, but not her son, passes mitochondrial DNA to her children. Because mitochondrial DNA is passed through the material line from mother to child, it contains information about a small number of a person's ancestors. However, since it passes from generation to generation with very little change, mitochondrial DNA is a rich source of information for deep ancestry going back thousands of years.
Good day everyone. Today, I'll be explaining one of the important part found in the mitochondria, the ribosomes. But to help us visualize this better, I'd prepared a short video presentation about ribosomes and its functions. When you look at the pictures of these plant and animal cells, you can see tiny dots on the ER and in the cytoplasm. These dots are the ribosomes, but why are they important to the cell? Ribosomes are small protein factories found in the cell. Ribosomes are found in plant, animal, and bacteria cells. The average eukaryotic cell has approximately 10 million ribosomes. If you take a look at this picture of a ribosome, you will notice a large subunit and small subunit, which both contain protein and RNA. In plant and animal cells, ribosomes are found floating in the cytoplasm or bound to the rough ER. Free-floating ribosomes create proteins to be used within the cell, while ribosomes attached to the ER create proteins that are either bound to the cell membrane or are used outside of the cell. Ribosomes use a process called translation to create proteins. In basic terms, here's what happens. mRNA or messenger RNA that has been created at the nucleus leaves the nucleus and travels to the ribosomes. Next, tRNA then travels to the ribosomes and reads the mRNA in triplets called codons. The tRNA has an amino acid attached to one end. As the tRNA reads the mRNA, the strings of the amino acid are put together in the correct order and creates a protein. And that would be all. Thank you for listening, and I hope that you have learned something from me.
In conclusion, the unique structures of the mitochondria are directly connected to their functions and cellular respiration. The outer membrane protects the organelle, while the inner membrane with its fold called cristae increases surface area for producing ATP. The matrix contains enzymes and DNA that helps break down food molecules to release energy. Because of these specialized structures, mitochondria can efficiently turn nutrients into ATP, which serves as the main energy supply for the cell. That's all for our topic. We hope this gave you a better understanding of the structures of mitochondria and how its function relates and cellular respiration, and maybe even inspired you to think about it in a new way. Thank you for listening.